Hello everybody. So after discussing the histology of epithelial tissue, connective tissue and muscle tissue, we've finally come to the last, fourth type of tissue, which is nerve tissue. It forms the nerve system. You know the nerve system is composed of the central nerve system, which includes brain and spinal cord, and the peripheral nervous system, which includes the nerves, either cranial or spinal, and the ganglia. The cells of the nervous tissue are neurons and the supporting neuroglia cells. And if you will ever hear terms like gray matter and white matter, then the definitions is as follow. The gray matter is accumulations of bodies of neurons, while the white matter is accumulation of processes of neurons and their sheaths. Like envelopes. Let's mention briefly the embryology and the embryologic origin, because it's relevant when discussing the organization and microscopic anatomy of nervous system. Let's have a cross-section to the human embryo, an early embryo that has the dorsal ectoderm that thickens in the neural plate, which starts to form a neural groove. This will be the neural plate and the neural groove in the center. Below we have the notochord, which helps to organize these morphogenetic processes, and the mesoderm, the dorsal aorta, they are paired at the first time, and ventrally there is the endoderm layer. So that's the transversal section through the human embryo. Here will be dorsal direction. Here is ventral. This is the mesoderm. These are paired aortae. So the left and right dorsal aorta. And that would be the endoderm. Sorry. Now, if we focus on this region, then there is a process called neurulation. When this groove becomes deeper and deeper, the neuroectoderm invaginates towards the notochord. The dorsal aorta below are fusing, by the way, into a single aorta. And this material here will become a special organ, and next step, so I'll mark it with different color. So this is closing of neural groove and invagination. So finally, the outcome of this process called neurulation is as follows. The dorsal ectoderm will close and this will become an independent organ called neural tube. Surface body, ectoderm, dorsal ectoderm of the body. This will be the neural tube. This material will become an important embryonic structure called neural crest on both sides of the neural tube. The neural crest has a great proliferation and migration potential. It will migrate into many parts of human body. So the neural crest will differentiate in structures such as spinal ganglia, and the autonomic nervous system, including sympathetic and parasympathetic ganglia, including the enteric nerve system. There are other structures such as the placodes of sensory cranial nerves, or the adrenal medulla, pigment cells, the melanocytes, some sensory cells of skin, Merkel cells, and other structures. If we go back to the organization of the neural tube, below which we will find the notochord cord and the dorsal aorta. Then the neural tube has a basal plate ventrally. It has the alar plate or roof plate. Dorsally, here is ventrally. Here is dorsally, is ventrally. And it has the sulcus limitans. And if you look more closely on the distribution of various functions in the neural tube, especially in the spinal cord, then here is the ventral direction, here is dorsal, as on our developmental schemes. Then the most ventral region becomes somatomotoric region. This will become visceral motor region. So the ventral are the motor regions. The dorsal are sensory region. This will be visceral, sensory, visceral sensitive. And this will be somato. Sensitive or somatosensory region.